recently to HVivo, but I've been there for pretty much 20 years. I was the first member of staff. We're now 350 people. My official title is Executive Scientific Advisor, and if anyone can help explain to me what that means, I'd be really grateful over lunch. But I've done challenge studies since 2001. I did the very first study, there you go closer to the mic. I did the very first study in 2001 in a hall of residence at the University of London. And it was on 8 volunteers, 4 of whom were friends of mine, because back then you were allowed to actually experiment on your friends. You're not allowed to do that anymore. 2 of them are still friends of mine. But there we go. So anyway, what is it that we do? And this isn't intended to be an overly promotional thing. It is a description of our history and what we've done in the past. So all of these slides are available anyway. But we've done over 40 clinical studies now. We've infected almost 2,000 people with influenza, RSV, or rhinovirus. We've got multiple proof of concepts. We've done vaccine studies, we've done siRNA studies, we've done small molecule studies. So we've done a huge amount of work, actually, and we've taken this model. So, get a volunteer, get them in a room, infect them with a virus, and see what happens, is fundamentally what we do. So, but yeah, it's, I hate it when there's not actually a mic here, but yeah, that's not working. Okay, so yeah, we've been doing it for a very long time now, and that's the most important thing. What we've also managed to do, though, and it's on this slide, is from those studies, we've manufactured the viruses that we use. We then actually take the data and the samples from the studies where we work with clients, and we're actually creating a biobank as well, so that we can really start to understand the interaction. Because we've got a volunteer, turns up, we infect them, and then we have bled about 700 mils out of volunteers over a 10-day period in certain circumstances, and that was with Duke University. So we can get a huge amount of samples, etc. One of the key points here is, though, and that was the reason I actually asked the question earlier, we're not the only people who do this. We have been the only people who have done this since 2001, but in the last couple of years, a couple of other groups are now doing this. And they're using the H1N1 virus. We chose not to do that. We chose to use the H3N2 virus because the CDC agrees that the H3N2 virus is the most significant virus. It's the one that has the greatest incidence. It's the one that has the greatest mortality and morbidity. So we actually, this wasn't a random thing. We didn't go for the sexiest virus. I get into trouble for about what I'm about to say, by the way, and I'm probably been told not to say it. But we didn't go for the virus with the shortest skirt. It wasn't the sexiest virus. We went to the virus that actually had most sense to actually manufacture. So, and this was the virus. And that actually is exactly in tune with what the CDC says. H3N2 is, the most significant virus, even allowing for pandemic H1N1. If you actually look at the numbers, H3N2 is the most significant virus. So basically, what do we do? Our aim as a group, and just to give you a bit more background, I'm not just a member of the company, I'm actually an honorary senior lecturer at the University of London, and an honorary lecturer at the University of Brighton, so I am a geeky academic as well. So I'm not just a corporate dude. But what do we do? What we're trying to do is we want to take what happened at the Common Cold Institute, which was set up in 1946, with the intention of accelerating research. And we've tried to duplicate that since 2001. So basically, this graph is basically intended to show you that instead of going out into the field, you can actually come to us or the other groups who are doing it, that's fine, and actually go, right, a challenge study is a better way of doing this. You're not going to get license on it. That's not going to happen. But what it is going to allow you to do is speed up your decision process on what dose of drug you want to use, timing of vaccination, or whatever that is. And we've done that in multiple cases now, and you can search us and you'll find it. So, so I mean, basically, this is exactly what I'm talking about. Somebody much cleverer than I has done some very nice graphics here, but basically what it's telling you is that we have actually gone from, initial start of a clinical study to a result within a year, which has then led them to be able to go right into a field study, and very successfully into a field study. So, right, I'm just going to shout. Okay, so there's a lot of regulatory support. You hear me okay? Yeah, good. There's a lot of regulatory support. The FDA issued guidance in 2011 that supported the concept of challenge studies. The World Health Organization gave out guidance about the use of challenge studies. Challenge studies, are not going to get a license. I want to be very clear. But what they are going to do is they're going to allow you to make a decision about what you do when you move to the next step. And that's going to save a lot of time and a lot of effort. And in addition, we get a lot of extra data out of it. So we get really good results. We get there faster. We also are having quite a bit of fun at the moment, actually, I have to say, as a company, because we actually consider it a case that we've spent a lot of time developing our viruses, and we consider them to be our viruses. So we consider them to be the case that we're actually licensing those viruses out to people. So, and why that's important to us is, not only are we actually helping our clients develop their drug and move it forward, we're actually geeky scientists. I'm sorry, that's just the case. I'm a geek, and at the University of London. But it means that what we can do is, from all the placebo samples that are left over after a clinical study, we can actually start to interrogate them and actually look for new things. So we look for biomarkers, etc. And that's what's good about the model. Not only do we help our clients and our collaborators to actually get their vaccine or their antiviral drug the next step along, we end up with data and samples that we can then interrogate, anonymously, of course. And I think that's hugely important, because that means that we're not a CRO, we're not a contract research organisation, we are not quintiles. We actually do real research behind what we do. So we can add a lot of additional info. I mean, personally, I've been doing it for far too long and probably should give up soon, and retire. But quite a few of my colleagues have as well. I mean, we've got a real bedrock of staff within the company who have been doing these studies for a very long time. So we've got that great experience that allows us to do study design. We've also have the benefit of being in London, and that actually works extremely well, because London's a big mismatch of people. Yes, it's like, we're all from all over the place. So so we've got this great mismatch, and that's really, really useful as well. So we can actually look at some interesting demographics as well. And in fact, we are looking at some interesting demographics at the moment. So the agents that we can use, I mentioned this. We started off with flu. That was the first one we did. It was a B virus back in 2001. As I said, it was, 4 of them were my friends. 2 still are, as I said. We brand the company slightly differently for recruitment of volunteers. We call it flucamp.com. And that's for the recruitment of volunteers. And the reason we do that is purely because those 2 friends had actually been watching Fat Camp on the TV. And so they then called it Flu Camp, and it stuck, and that's our brand that we use to recruit volunteers these days. So, but, that was our first thing, flu. But we've done rhinovirus, and we've done some brilliant stuff on respiratory syncytial virus, and looking at siRNAs, etc. Sample size, big thing, actually, because how you power a study is extremely complex. And we've, because we make it a condition of any agreement we go into, that the placebo data from earlier studies or that study is available to actually power a study. It means that our future clients can share, it's all anonymous, but our future clients can share that data to power their study to make sure it's the most effective study it can be. And therefore we put the least number of people at risk. Because at the end of the day, anybody in a clinical trial is at risk. So we take all that data, we collect it together. I get terribly excited about it, but there you go. But it's like it brings it all together, and we're able to take that data, leverage it to design a better study for the next person on the line. I think that's entirely the way this sort of thing should work. And we do a variety of different types of studies, different IMPs. We do vaccines, we do prophylactic IMPs, we do post-infection IMPs. So we've got quite a variety of things we do. And actually I've already covered this. I always do this when I do talks. I always jump ahead of my own slides. It's like, but there you go. But this is the point. It's like, the beauty of the challenge study is that you can actually define your next study down the line, and you can refine it. And that's exactly what the FDA said in the earlier slide, and it's exactly what the World Health Organization said in the earlier slide. And I think that's the key message, if there's any message that I want to get on. Bit of our history. Common Cold Institute in the UK, I'm slightly surprised that I don't have a slide of it, was set up in 1946. It was the old Harvard Institute, hospital rather, at the end of the World War. And the Americans legged it out the country and left it with us. Well, it was quite fine, because the Medical Research Council used it for about 50 years. I think it was 1997, 86 actually, when they actually closed down. But for years and years and years, they did loads of research there, and they went through about 20,000 people over all of that period of time. We've almost got to 2,000. I was so hoping that I was going to be able to update this slide to actually say, yeah, we got to 2,000. So they did 20,000 people infected. We've almost got to 2,000 now, and we've managed that in a decade. So we have a hell of a lot of experience of this. But all of that data, all of those samples, they're all going in there, and, that experience. And one of the big things is that we've kept a lot of the staff. So all that experience is there. Quickly, I'm just going to mention the H5N1. We haven't quite got there yet. We have a virus. These are our viruses. I mean, there are other groups who have other viruses, which is fine, but these are our viruses, and one of the ones I really want to get done is H5N1, because it's a very attenuated strain. But we're confident it's going to be safe, and we think it's actually an important virus to actually get the model developed for us. So, on the matter of attenuation, generally our viruses aren't. It's a myth to believe that most challenge viruses are attenuated. They're not. They're not wimpy. They're not attenuated. All it is the myth comes about from the fact that in the States, back in the 90s, they used Atexus H1N1, which was a mild virus. And so people assumed that generally challenge studies were wimpy viruses. It's not the case, and we've got a lot of data to support that. So very quickly, because I'm conscious I'm probably running over my own time. So one of the things we're very good at, and bearing in mind I've got the luxury of not being involved in the general operations of things, so it's like other people get to do that, but it's fair to give them credit here, because we have an extremely good recruitment process. So when you think that about 1 in 10 or 1 in 20, somewhere around that, number of volunteers who actually phones us up ends up in a study, you can imagine how that funnel works, that it's extremely difficult. But we've got a great team who can actually do it, so it's really, really good. We've got facilities, we have a 24-bed quarantine facility. All of the rooms are en suite. They're probably better than my room at the moment. So, really, really good, really good. Not an open ward, and I think that's really important, because if you get cross-contamination, Sir Charles, actually, who did some stuff very early on in the 1940s, one of the big problems he had was that he used Bart's medical school students. And the big issue was they went out and then they got infected with something else. So how on earth did you know what you were measuring? So that's one of the reasons we've actually been really strict, and we've got individual rooms, and they're all en suite, and extremely strict control procedures in place. And the staff, obviously, are very well versed in infection control. So this is just, I will admit, I wouldn't have put gold standard on it, but hey, somebody did it. So this is a really good example, though, of a study. This was a study we did back quite a long time ago. We published on it. And it was the first demonstration of an siRNA. So a really, a novel anti-infective. And since then, we've published with Alios and Gilead, again from the model, really good data. So really nice stuff. So the data has been shown to be very, very robust, and then has actually led into subsequent studies. So I think that's really important. Also, it's one of the things is it happened actually on 2 occasions, but for a lot of companies, smaller companies, it's important for them to be able to move on to their next step. Most companies are going to want to be acquired, list, whatever. Even academic groups want to move on. And it's a good example that after we finished our study with Alios, they were acquired by J&J, which are an interesting group to work with, but I'll leave that to another discussion. So, I'm getting the paper. Okay, all right, I'll move quickly on. So anyway, as I said, model really good. John Fry is a brilliant guy, actually, at Alios, now JJ. We published our work, and we have a very strong policy on this, by the way. It's like we don't try and hide anything or anything like that. In fact, it's part of our contracts we go into with our clients that we say you've got to go for the concert principles, which is that you publish. Whether it's positive or negative, you publish. And that's pretty much repeated. And this pretty much repeats everything. And by the way, that's the Flu Camp branding that we have, which actually is hugely popular with our volunteers. And there is #FluCamp that you can now follow on Twitter. And it is actually quite amusing to I actually do have it on my screen at home when I'm working at home. And it's like, I love seeing the little phrases that are actually coming up on the screen. So, repeating myself here, this is the facility, en-suite rooms, very tightly controlled. We did not just convert an old phase 1 facility. We built this from scratch, and to its absolutely exactly what we wanted it to do. I'm going to skip that. And again, this just really repeats what I've said already, that we've published. We have a strong belief in publications, and we've had multiple proof of principles. Very proud of that. In fact, I'm working on a paper at the moment, which I was up late last night working on. But we've got multiples from everything from Nature Medicine to vaccine to +1. So it's like lots of stuff that we've done. We had the first proof of concept of a DNA vaccine, a T-cell vaccine, lots and lots of stuff. Very, very quickly, how much? I've got 0 minutes, apparently. That's okay. Very quickly, this is slightly repeats what I've already talked about, and I will do this very quickly. But we manufactured recently a new virus, as I said. We chose to do the H3N2 route. We carefully, very carefully selected it. And we did a lot. We put it all through this entire process, and all of these slides are available if you want them. But the key thing here is that we did a huge amount of adventitious agent testing, GLP testing in ferrets, and we will be publishing that very shortly. Then we did a characterization study in humans, and that's actually also about to be published. And now that's all been diluted down, that's in our freezers, and we've got enough stock to infect half the population of London. And by the way, I discovered you shouldn't say that when you're on the phone. People apparently get annoyed when you're sitting next to them on the train. I don't know why. Anyway, study design, basically, if anyone wants to talk to me about this later. But, there's a whole process. Volunteers are admitted to the unit. We will screen them. We will check whether they've got antibodies to the virus we want to infect them with. We'll then infect them after we've made sure they don't have any other virus. And then somewhere around 10 to 14 days afterwards, after taking lots of blood and lots of nose samples out of them and measuring lots of virus out of them, we will actually discharge them, and then we'll follow up with them later. So, all right, I'm done. Okay. There you go. Any questions?
Dr. Bizon a minute ago showed a profile kinetics in which PCR could be measured for virus out to, I don't know, she would know better, 10 to 14 days.
Yep.
I hope you're not allowing the units to be departed until such time as they're PCR clean?
No, we actually, as long as they're asymptomatic and there's no virology, we have had people being discharged when they are PCR positive. But we will use a DFA or something like that to clear them at the end of a study. But as long as they're asymptomatic, the risk of them transmitting virus is minimal.
Have you ever had the opportunity to recall volunteers to look at durability of the response to prior infection, or how their pre-immunity might be affecting their response to infection?
Yeah, we're actually doing that. So yeah, absolutely. I mean, it would be fascinating. I mean, one of the big things I'm really interested in at the moment, and actually, we've got another paper submitted at the moment, is looking at mucosal IgA. And somebody mentioned it earlier, and I can't remember. I do apologise. But we've got a really nice graph that actually demonstrates how mucosal IgA and serum IgG interact. to reduce the symptomology of volunteers in a challenge study. So, it's... Anyone else?